Hey gang, this is the Bacteria UV Exploratory Lab, and in this experiment we're going to take a look at a strain of bacteria. We're going to expose them to varying amounts of time to UV light and see its impact on the amount of bacteria that grow. We're looking at the number of colonies that grow of bacteria, and you'll know the colonies basically are look like clumps of bacteria. When you submit your data, you're of course going to submit it to a form. So you put in the number of colonies at these different amounts of time, 0, 30, 60, and 300 seconds. And then that data is going to get collated, and it's going to be the average number of colonies is going to be calculated as published data. So I want to talk you through some of the materials and the sterile techniques. When you're growing bacteria, you have to use what's called sterile techniques because you don't want the wrong bacteria to grow. So there's bacteria everywhere, on our hands, in the air, on tools, etc., etc. So if we're doing an experiment on a particular strain of bacteria, we don't want a different strain of bacteria to contaminate our workspace. So whenever you sterilize your area, you're gonna have to get some alcohol and rub down the table, pour some alcohol on it, use a paper towel. You're going to have to get your bacteria onto a petri dish, and you'll see there's agar on the bottom. That's the food for the bacteria, and we have to get the bacteria on there. But if we were to open the lid of this petri dish, then bacteria from the air would contaminate the petri dish. So we're going to use something that's called a clamshell technique, and that's when we slightly open the cover just enough to get a pipette or spreader beads, etc., into the petri dish. So we're going to use a lot of disposable materials. We have a pipette, and they come packaged in its own container, its own wrapper, excuse me. And when we open it, we want to open it from the back so we don't inadvertently put our infected hands on the part of the pipette that's needed. What we do is we need to spread the bacteria throughout the petri dish. So we're going to either use glass spreader beads. So we have sterile beads, and we're gonna clamshell, put the beads inside, then we're going to shake the petri dish to get the bacteria to spread, or we can just use a sterile spreader, this blue handle thing, and we can use that to spread it. Once you have all of your plates finished with bacteria in it, then it's time to expose the bacteria to the UV light. We're going to start with the 300-second group and make our way down. That will give us time to make any last-minute corrections. And the very most important thing for this lab is that you should never look directly at the UV light. We're going to wear goggles, but even with the goggles, you should not look directly at the light. So that was the UV bacteria lab. Thanks, gang.